My name is Mark Lucanic. I am a co-founder of Gerostate Alpha. So today I'm going to tell you a little bit about GSA. Specifically, I'm going to tell a bit of the story of what led to GSA and briefly part of what we do. So when I first got into the aging field, I was coming from the perspective of a developmental geneticist. And I'd been doing a lot of genetic screens, essentially to dissect out what genes function to control aspects of development and how they function to control those aspects of development. When I was looking for a new project to join, I chose the aging field. I was really interested in lifespan determination and the idea that, what exactly determines who lives how long. And so when I was looking around, I ran into a pitch by Gordon Lithgow, one of the three co-founders of Gerostate Alpha. And he was looking to do a screen for compounds, drugs, small molecules that extend lifespan. And he was working in this model organism, this small worm I'll tell you a bit more about shortly. And so I knew that, identifying drugs that could extend lifespan was science fact, not science fiction. In part because Gordon, along with our third co-founder, Simon Melov, had shown in the early 2000s, in fact, they were the first to show that small drug-like molecules could extend lifespan in this same model organism. Subsequently, it's been shown that drugs can extend lifespan in many organisms, including mice. So I joined the lab and I did the screen. At first, we set up and I spent a lot of time figuring out how to culture these worms in a format that was appropriate for small molecule screening through their entire lifespan. So there's the rub, right? In a lifespan screen, you have to have them viable the whole time. And anyways, we came up with a system doing that. And here I'm showing you a schematic of that. So we use these 96 well plates. Each well contains a population of these worms. And each well also contains a unique drug. So I screened 25,000 compounds and identified several that extended a lifespan. On the right, I'm showing you one of these drugs, NP1. And here I'm showing you the survivorship curve of a population. Okay, so here's a control population, untreated, and they 50% of this control population is alive at day 18. So we say they have a median lifespan of 18 days. Contrast that with when we treated them with the drug, the median lifespan was more like 22 days. So, like a 25% lifespan extension. So it'd be like if a human had a median lifespan of 80 days, that would be like 90, plus median lifespan, 20% change. So, so we did the screen, published that screen as well as the method. I encourage you to read more about it if you'd like. But really what I came out of that, so we did the screen, that was and then I spent the next, several years describing exactly how that drug extended lifespan, what the underlying mechanism was. So, worthwhile and all that, but ultimately, if you want to have an impact on human disease and aging as we do, it's perhaps not the most efficient way to go about it. So while I was doing that, I was also involved in this other project, the Center-Hepatitis Intervention Testing Program. Really interesting program, funded by the National Institutes of Aging. And I definitely encourage you to read more about that as well. So in that program, the NIA was looking to test interventions, mainly drugs so far, for two properties, essentially two properties. those properties being reproducibility. So they want to do, they wanted to be clear that these drugs had this effect independent of where these tests were being done. So the program is a consortium of 3 labs across the US. And so we would test these drugs in each lab would test these drugs and see if the result was reproducible. Also, we were looking for robustness, so whether these drugs would work across genetic distances. So to get at that, we used, again, we're using these roundworms, but not just a single species, which is the standard. Normally you would use this lab-adapted strain of C. elegans. So we had 3 species in there. In fact, we had 22 strains, many of them wild isolates. And so we were looking for robustness, so things that would work across these genetically very different species and strains. And that's important, particularly if you're looking to move these drugs into more complex organisms where these experiments are harder to do and it's more expensive. Then the odds of them working there should be increased if the target's conserved. You can see the target is conserved if it works across genetic distance, likely to be conserved. Okay, so here is some of the data from the CITB. So here I'm showing you survivorship curves again. So these 3 curves come from 21,000 individual animals. And here I'm presenting them by species. They could also be graphed by strain. And the trend lines, when I graph them as strains, is similar to species. So the reason I'm showing you this is part of the reason these experiments are nice is that you have a lot of diversity of mortality dynamics shown here. So we've got these 3 species and they have different lifespans. And not only that, but they die so they have different median lifespans. So here, these 2 have different medians, but they have similar max. These 2 have similar medians, but they have different max. So that's really interesting, but perhaps more interesting is the fact that when we did these experiments, I selected 10 drugs initially to look at in these strains. What we found was that often we would see a drug that would affect a single strain, or in this case, a single species, but not another species. So we found exactly what we thought in that sense. I mean, it's what you would expect is that some drugs were better across species and strains, across genetic distance, than others were. Okay, so around that same time, Simon Melov, one of our co-founders I mentioned earlier, was also working in aging. In fact, he was at the Buck Institute, as Gordon and I were, and he was working in a different area of aging, and in fact, a whole different model. He was working in mice, and what he was doing is he was trying to quantitatively measure what happens as we age, or as mice age. So what he was doing is he was taking middle-aged mice and then measuring aspects of their physiology as they advance into old age. He was looking at fat levels, bone function and morphology, cardiac function, pulmonary function, even things like muscle force. And he was looking at how these decline with age. That's what happens when we age. These systems decline. So he's doing more than that, though. He was actually looking for, and so when you look at these over time, you see this negative slope of the function of these physiological features and systems. He was also looking for interventions that could decrease these slopes, so improve aging. One day, Simon came to Gordon and I and suggested that we could combine all of our projects and form a company. And the real draw there is that by forming a company, we were much more likely to do something that directly leads to an improvement for humans, for society. This was going to be the way where we were going to get something that was going to be used to improve human populations. We were going to get a drug or something into people. We could do everything faster and streamlined. And essentially the sort of makeup of Gerostate incorporates those 3 projects that I was just talking about. So we now use lifespan as the primary entry point for our drug discovery platform. Early on, we demonstrate robust effect across genetic distance, and they have to slow some feature of what we're testing for ultimately is slowing a feature of aging in vertebrates. Okay, so here's a like simplified schematic of our screening aspect or our platform. So we screen for lifespan, specifically for things that promote lifespan in this simple invertebrate, Cinerabditis, and then we move these into mice to test aging and all these physiological features where we quantify their effect on the aging of tissues. So we're screening both lifespan and aging. Okay, so most of our platform is based on drug screening. So what do we screen? We screen approved drugs. So we just actually did a small, relatively small scale screen of off-patent drugs, something like 1,200 drugs, and we identified some that extended a lifespan. Our simple model, we moved them in some other subset of those into mice, and identified one that had a significant effect on improving bone morphology with age, and in fact, restoring some aspects of bone morphology to more youthful levels. Also, interestingly, had a significant impact on improving muscle strength with age. So we're still continuing to look at that drug in follow-up experiments. We'd also like to start screening natural products, and we have some plans for moving into natural products. But ultimately, where we think most of our impact is going to come from are on identifying entirely new aging pathways by exploring chemical space, utilizing diverse chemical structures in our primary screens. So we've done some of that. So I already did with GSA a 60,000 compound small molecule screen where we looked at synthetic chemicals, libraries of synthetic chemicals that were selected for their diversity and drug-like properties. So what I'm showing you here is when we were doing that screen, I just took an image, a picture of our incubators full of these screening plates. So they're full of these 96 well plates I talked about earlier, and they're incubating, waiting to compare to control, to screen for which compounds are effective at extending lifespan. I calculated this at one point that there's about 1 million individual worms in these plates, and there's about 20,000 drugs on those worms. Okay, so we did the screen, and I identified tens of, just over 30 promising candidates that have now been validated. So when I say validated, I mean that we have repeatedly demonstrated in multiple different assays that these drugs extend lifespan. Some of them are very potent. So here's GSA 7, again, a survivorship curve. We got a median lifespan in the control of about 12 days, 13, maybe 15 days. And GSA 7 here is about 20 days. So this is a lifespan extension of like 30 to 50%, very potent. One of the most potent, if not the most potent, chemical to extend lifespan that has ever been in my hands. So we have others, quite a few others, as I said. Here, what I'm graphing is this is normalized to control. Each point here is a individual plate assay. As you can see here, GSA 4, no effect at a low dose. As we increase the dose, we get the significant and potent effect on lifespan. The same is true of these other drugs. We have a lot of candidates. Now we're ready to move these candidates that extend lifespan into our mouse, our vertebrate aging assays. So that is what we're aimed at right now. We also, in the future, want to screen more compounds. I personally would like to screen 500,000 small molecules. I think I can do it in under a year and a half. We'd like to look at more known drugs. And we have plans to move into natural products as well. So that's all I have for today. Thank you so much for listening, and goodbye.